Hi, welcome back to 7th grade life sciences. This is our video note set for day 4 of Cell Theory and Life 101. So just a reminder, we need our blue lab notebook every day in class. Bring that back and forth to school, please. And every once in a while, you'll have to actually leave it with me, so you won't be bringing it home. But we'll be filling out things in that blue lab book regularly. So today, we've got some warm-up questions. Quick question. What is the name of the smallest parts inside a cell that have a specific job? They're all called organelles. And what type of cell is shown over here? I will always have shown you cells before in our classroom before I ask you to tell me what something is, unless it's something generic like, do you think this is a plant cell or an animal cell? We have seen this animal cell before. It is frog blood. So what organelle is number 1 pointing to? That blue solid center structure, or that blue dark structure there, that's going to be the nucleus. All right, so today we're starting out with a 6th grade 6-minute review. Remember, there are materials from 6th grade and 7th grade and 8th grade on your 8th grade SOL. So we will continue to review things that may be showing up as a problem or an area where there is room for growth. All right, so today's question is, what are the phases of matter? Sometimes you might have heard that as states of matter, and those would be solid, liquid, gas. Sorry. There is a 4th one, and some people remembered this in class, and some people weren't sure that they remembered it, but the 4th one is going to be plasma. Mostly we're going to talk about solid, liquid, gas at this point. So I asked everybody to kind of remind me of what they remember. Tell me what they remember. Solids hold their own shape. They don't change shape as you move them from one location to another. So solid is going to hold its shape, and it has a fixed volume. So like a pencil or an eraser or your desk or your book. Then liquids, they actually take up the shape of the container, and they have a fixed volume. Their volume doesn't change, but they do take up the shape of the container. And so you might think of putting milk in a glass, and you realize if you put milk in a glass that's a tall, skinny glass, it's one shape. But if you pour that exact same amount of milk into your cereal bowl, it's going to be a completely different shape, but its volume stays the same. And then a gas is going to take up the shape of whatever container you give it, and also it'll take up the volume of the container. So it expands freely. And here's my notes again. A plasma is sort of basically a hot gas that has more energy than all of the other 3 states of matter. And so we will probably not talk about plasma very much, but plasma can be found with lightning and sometimes with things like maybe a plasma TV or something. It's an excited state of matter. All right, so we talked officially today about physical and chemical changes of matter. So physical changes, that's going to be something that's reversible, mostly, that can be undone. Sometimes that's harder said than actually performed. So also it can result in a change in size or shape, but the material is the same material after your change. It might look different. It might be in a different form, even a different state of matter. So if you had ice and you melted it or it evaporated, but it's still exactly the same molecules, excuse me. Those are physical changes. And then chemical changes are going to be things that are irreversible. They can't be undone. We're changing molecules to make something different from what we started with. So a big example that we know of that we've talked about in class is when we do photosynthesis, we take molecules of carbon dioxide and water, and then they are exposed to chloroplasts and energy from the sun. There's a chemical change, a chemical reaction, and what comes out is something completely different, right? We end up with sugar, glucose, and oxygen, completely different molecules that come out of that change. You also might see changes in color, or a chemical change could have an odor, or it could make bubbles when there were not bubbles to start with, or even an explosion. So I have a little video for you And let me set myself up for this. All right, so if we want to think about physical versus chemical changes, what if we're talking about ripping a piece of paper? That one's going to be, think about it first and see if you can answer the same as me, physical. If we are mixing vinegar and baking soda, if you haven't done this, you can just do a little bowl or a little cup and put some baking soda in there and pour vinegar on top. It will fizz and make bubbles. That's a chemical change. Lighting fireworks, we see explosions, we hear explosions, we can see colors, right? That's going to be chemical. And then dissolving salt in water, that'll be a physical change. So that is reversible. You can actually let the water evaporate or use you can use a distilling apparatus. You can get the salt back out. It's not easy, but it is something that you can do. When you dissolve salt in water, you have salt and water that you can separate back out again. All right, so just a couple more. If we are baking a cake, so think about this one. If we're baking cake, that's going to be a chemical change. That is changing the chemical structures after we've been in the heat of the oven. If you're scrambling eggs, when you scramble those eggs, that is not a chemical change in the beginning. It's not a chemical change until after it's been baked, those eggs in that cake. Okay, making trail mix out of peanuts and M&Ms, that's just going to be a physical change. And ice melting in your drink, that's a phase change or change of a state of matter, right? So it's still water, it's just whether it's frozen solid water or whether it's liquid water. So that's a physical change. All right, so that was just a quick reminder about physical and chemical changes, and you'll see that come up on your exit ticket. All right, so today you will be observing mostly plant cells. We actually observed animal cells last class, and we decided not to do any new animal cells on today's class. We will observe animal cells in our next class, though. So we observed plant cells so that we can think about the animal cells we've already seen and the plant cells we're looking at now, and try to make a note of differences between plant and animal cells. And we'll know that we got it when we can identify those similarities and differences between plant and animal cells. All right, and so you need your blue book in order to do this next part, and you'll probably need to be in the classroom for some of this as well. So if you missed class, come on by during a Falcon Focus. Right now, though, this one you can do without being in the classroom. I asked everybody to write out the 3 parts to cell theory on the top of page 9 or maybe on page 10 in your blue book. Those are places in the blue book where we actually have some extra white space, and you can write some extra notes down and they'll be right there with you. All right, so cell theory, we know that we had some scientists talking about cells. Sorry, I don't know how to make that go away for us. And so they came up with 3 basic parts of cell theory. Number 1, all organisms are made of 1 or more cells. And number 2, cells are the basic building blocks of life. Number 3, all cells come from existing cells. And we know that we had different scientists helping us understand these things. So I just want to make sure that you are committing that to memory. Make sure you know those 3 parts. You can think of the ABCs of cell theory, if that helps you. So the ABCs of cell theory: A is all organisms are made of cells. The B is the cell is the basic building block of life. And C, every cell comes from another existing cell, or cells divide to find new cells, to create new cells. All right, so please do make sure you are committing that to memory. Remind yourself kind of every day so that you can list those 3 parts on your own. So, reminder about some of our scientists. You remember that we talked about Hooke. He was the first guy, I believe it was 1655, and he looked through a microscope at cork. So he was looking at basically dead bark, and he contributed to the whole idea of cells because he came up with the word cell. He's talking about the shape and what he saw when he looked through that microscope. That's the extent of what he offers us, basically. And so after he looked at those pieces of bark, the cork, he was communicating a little bit with Van Leeuwenhoek, And Van Leeuwenhoek scraped some stuff off of his teeth and looked at that under the microscope. Leeuwenhoek actually saw live cells. So that was 1655, and around then, the first two guys. And about 150 years later, we have Schleiden in 1838. And he sees that all plants are composed of cells. And nearly at the same time, in 1839, Schwann decides that all animals must be composed of cells. So we've got Schleiden and Schwann. And then finally, we have Virchow. Just a little bit later, 1855, he sees cells divide and go from being 1 to 2 cells. And so he says cells can only come from other cells. All right, so we looked at frog blood, and I meant to change this slide. So this is true. We did observe. I can't move this. There it goes. Okay. We looked at frog blood in our microscopes, one of the classes recently. And so that was an animal cell. Frogs are animals. So today we're going to be looking at plant cells. And so we're going to look at Allodia. This is Allodia. This is Allodia without a microscope. This is Allodia with a microscope. And we're also going to be looking at onion cells. So we're going to look at onion cells. Most of the onion cells were cells, the slides were slides that I prepared in the morning. And so I actually cut up an onion and peeled off layers and used iodine, which is kind of a yellow substance, to stain it. So when you look at the slides, sometimes you're going to see air bubbles. That's an air bubble. You don't need to focus in on the air bubble. Look at some other parts of the onion cell as well. Okay, also, it all depends on how well the nucleus takes in that iodine stain. So on these cells, you can actually see the nucleus. It's kind of light, but you see these darker spots. That's going to be the nucleus of your onion cell. You can see really clearly on our cells in our classroom this rigid outer structure. Onion cells are practically hexagonal. When we were just looking at Allodia, these are actually, they look more rectangular. But the onion cells, they kind of have a hexagonal look. But you can definitely see the structure of those cells. When you are looking at your cells in the microscopes, you're going to be looking for the cell wall, which is this outer structure. You're going to be looking for cytoplasm. On this cell, it's this kind of light green or light gray. It's like a jelly-like substance that's in the middle of the cell. You'll be looking for the nucleus, and the nucleus is sometimes harder to see in our Allodia. But you might have to focus up and down a little bit using your fine adjustment knob. And then, let's see, we'll be talking about cell membrane. So if this is my cell wall, this darker structure here, the cell membrane is on the inside. You can't really see it, but it is on the inside, just inside that cell wall. So I will show you some more examples coming up here. In our onion cells, you're going to be able to see the nucleus, the cytoplasm. You can see that there is a cell wall. That's very obvious. And I'm going to say, please understand that there is a cell membrane just on the inside of that cell wall. Every single cell has a cell membrane. Plant cells also have this cell wall that helps give it structure. All right, so we're going to be looking at two different types of cells. We're going to look at Allodia cells and onion skin cells. And you're going to be making a drawing in your blue lab books on page 11. So you want to definitely do that. If you weren't in class, you'll come back during the Falcon Focus and take care of it. All right, I need to tell you some of the information about some of these structures and organelles that I've been mentioning. And we'll get into this more because we actually have more organelles, but just since I'm making you label these things today, the cell membrane, it's found in both plant and animal cells, and it provides a boundary between the inside and the outside of the cell. It actually does allow some things to pass through it. So it's almost like a big baggie, and you've got all your cell stuff inside that big baggie, but it's a baggie that is partly permeable. It allows certain things out and certain things back in. And that's important because in order for a plant cell to do photosynthesis, for example, in the chloroplast, we have to have carbon dioxide and water, right? That's how we do photosynthesis. Well, we can't have that unless the cell membrane allows carbon dioxide and water to come in. And then once it creates sugar that the cells need and oxygen, that's going to allow the sugar and the oxygen to transport itself out through that cell membrane as well. Everybody has a cell membrane, plants and animals both. They all have a cell membrane. And it's hard to see. It's just going to be containing the stuff inside the cell. Right now, a cell wall is only going to be in plant cells, not in animal cells. And it provides structure for that cell. Okay, so we also have a nucleus, and the nucleus has all the DNA, or the genetic material, the instructions. So sometimes people will call that the control center. And then we have cytoplasm. Cytoplasm is just kind of this jelly-like fluid. It surrounds all the organelles. So if I put a whole bunch of stuff in my pretend baggie, my cell membrane, it's not just hanging out there in space. It's actually moving around in this cytoplasm. All right, so we've got our two cells that we're going to be looking at, animal cells and plant cells. Plant cells have this cell wall, but plants also can have chloroplasts. And so this is a really good example of the chloroplasts. It's the green disc-like shapes that are where photosynthesis occurs. That's only going to happen in some plant cells. So this is a plant cell. And this dark outer, really clear structure that we're going to look at is the cell wall. Inside the cell wall is a cell membrane. And then, just wish that wasn't in the way all the time, you know that these little green discs are chloroplasts. Today, you're not going to be labeling the chloroplasts. You're just going to be labeling cell wall, cell membrane, nucleus, and cytoplasm in your cells. And so the cytoplasm, again, it's that jelly-like substance. It's sort of light greenish or grayish or clearish in here. It just surrounds all the other organelles. So you're also going to be trying to focus in and out, and you're going to try to find the nucleus inside your Elodea cells as well. All right, so here's just a drawn image of a cell. We have the nucleus, which is usually a darker, more obvious structure that's inside the cell. We have a cell membrane. That's going to be this light blue right here. It is, if it's in a plant cell, it is just inside the cell wall. If it's an animal cell, there's no cell wall at all. It's just going to be the outer part of your cell. Okay, cytoplasm is that jelly-like goo in which everything else is hanging out. And then this dark outer edge here is only in a plant cell. That's the cell wall. And the chloroplast is only going to be in a plant cell as well. Okay, so those are only in plant cells, the cell wall and the chloroplast. But everybody has a cell membrane. Most cells are going to have a nucleus and cytoplasm. Okay, so you're going to be observing the two types of plants today in class, onions and Elodea. And if you would, please look in your lab book on page 11. And, let's see, sorry, let me back myself up. Please look in your lab book on page 11. So it said prepare a wet mount slide of Elodea. You're not doing that. I actually have prepared slides that we have purchased for the Elodea. So you can put a little X over there. Don't, wreck up your book, but just a simple little X because you don't do step 1. Also, you're not going to do step 3. So on placing iodine, I actually placed iodine on the onion cells for you. And again, we have prepared and purchased Elodea cells. So the only thing you're going to do is step 2 for the Elodea. Read the directions about what you're going to be doing. You're going to focus on one layer of cells at the highest magnification. You're going to draw the outline of 6 or 7 of the cells. So you're only going to have 6 or 7 represented in your view. And draw the contents of one of the cells. you're going to easily see chloroplasts, but you're going to try to find a nucleus as well. And then you're going to title this drawing. If it's Alodia, you're going to write the word Alodia down here on the line. And if you were at 400 times, you're going to write 400 times there. All right, so make sure that you have read those instructions. And then just to be sure, you're only drawing 6 or 7 of the cells. Okay, and you've got to write down which cell you're observing and what is the magnification. So previously, in some of the classes, we were able to look at cork under 400 magnification, and today you're going to be looking at Alodia under 400. Probably you're going to look at your onion cells under 100 magnification. You can look at the higher magnification and see what you like better. Remember, these are self-assessment questions. When we are making a drawing, you're going to use pencil. You're going to use up an appropriate amount of space for what you are seeing and how big it is in your field of view. But you don't have to fill the entire field of view with drawings. You can do 6, 7, 8 cells. You don't have to draw, 72 cells. That's okay. And you're going to show some attention to details. So you want to include natural colors of the object if you can. You want to make sure it's similar shape to what you're actually seeing, and if there's a texture or anything. And then make sure you label using horizontal lines that go out to the side. And don't use any arrows, don't cross your lines, and use proper spelling. Okay, at the bottom, you're going to have the name of your object and the power. All right, so the thing that you'll do after you do your Alodia cell, then you're going to actually be making a record of your onion cell, also on page 11. So 6 or 7 of the cells, that's what you're going to draw, and then draw the contents of just one of the cells. On the onion cells, you're going to pretty easily see the cell wall, and it depends on the day how well the nucleus will take that iodine dye. Okay, so again, that's kind of what the onion cell is going to look like. All right, so I gave everybody about 15 minutes to try to do those cells, and then we came back to our desks. And I asked everybody to say, in general, what they observed that were similarities and differences between Alodia and the other plants. And so mostly, if we're looking at plant cells, we're going to see that outer structure, that cell wall. But we did not see any chloroplasts in the onion cells. So if you think about it, an onion actually grows underground. The bulb of the onion that we eat grows underground, and it has green stalks that come out of the ground. So it's the green stalks that are going to have chloroplasts, right? Because the green stalks are exposed to the sun and they're going to do photosynthesis. That bulb of the onion, it's underground. It's not getting any sun. It doesn't need any chloroplasts because it's not doing any photosynthesis. All right, so of course, always remember when you're done, double check that you have taken your slide off the microscope, revolved to the red low power, and you put your stage in the lowest position. And especially at the end of the day, if you're 7th or 8th period, you're going to put the cover back on your microscope. Make sure the light is off, and that's awesome. Everybody's been really good about that. Okay, so we did a Pear Deck a little bit in class. Let me see if I can go through the Pear Deck on this video. All right, so here we are. I'm going to do this Pear Deck just like a slide deck since you're not actually doing the Pear Deck yourself. I asked everyone to write at least one of the 3 parts of cell theory. And remember, that's going to be, if you want to think of the ABCs, all living things are made up of cells. And then B, the cell is the building block of life. It's going to be the smallest living thing that we know. And C, all cells come from other cells. Okay, and then this slide, I asked people to circle things that are made up of cells. It's going to be bacteria and fungi and plants and archaea and animals and protists. All of the living things are made up of cells, but our abiotic factors are not. So it was nice and organized for you to look at there. And then in your Pear Deck, ooh, no, will it let me see it? Down at the bottom there, you can see there's a gray star and a green circle and a purple square. So I asked everyone to move the green dot to the box showing the chloroplast. So the box showing the chloroplast. Get all my boxes showing up here. So this is a chloroplast right here. Nope. What am I saying? I want the chloroplast, the cell wall, and the cell membrane. So this is our chloroplast. One of the green discs goes right in here. And then the cell wall is the outer cell wall. That's that purple box. That's going to be your cell wall. So I had people move the square box to the cell wall. And then the inside cell membrane, that's going to get your gray star. So you would drag your gray star there if you had been doing the Pear Deck. All right, then it was multiple choice. Plant cells are different from animal cells because they have a cell wall. Only plants and some protists can perform photosynthesis. So what is the name of the organelle where photosynthesis occurs? And hopefully you remember this from previously, but also from our past couple of classes. We have as options nucleus or cell wall or cytoplasm or chloroplast. The organelle where photosynthesis occurs, that's the chloroplast. That's the place where photosynthesis occurs. Okay, and then this is an onion skin cell that does not have iodine dye. That's why it's kind of clear and blue. And so when you looked at cells in the classroom, you might have seen sections that didn't get as much dye that were more of this kind of whitish and blue color. So in this case, I asked people to draw a line from the cell part on the right to its location on the cell that's indicated. So your cell wall is going to be the outer part. Let me back myself back up. Outside of the cell is going to be your cell wall. Your cell membrane is going to be on the inside. So you can see the cell wall pretty easily. You just know that the cell membrane is here when you're looking at a plant cell. Cytoplasm is going to be the white jelly-like stuff. And then your nucleus is going to be your dark center structure right there. Okay, and then because you did this in class, I asked everybody to double check on page 11 and just make sure that your cells looked pretty appropriate. Remember, you were only drawing 6 or 7 cells, so you probably don't have this many, but you want to have a cell membrane listed and a nucleus and a cell wall on the outside and cytoplasm. Double check that you've got everything labeled properly as well. Like I said, for the most part, we did this on 100 times because you could see plenty on 100 times and you didn't really need to go to 400. If you drew yours at 400, though, you need to make sure that it shows up in the size that they appear to be and that you've written down 400 instead. Okay, so what do these plant cells have in common? They're both plant cells. They both have a nice structure from that cell wall. And they definitely both have cytoplasm, a cell wall, a cell membrane. And you can definitely see the nucleus in the onion skin. It's harder to see the nucleus in the Elodea. So those are some similarities. And if we want to think about differences, well, our Elodea is green and our onion skin is not green. And our Elodea has a bunch of stuff inside each one of these cells that's very visible. The onion skin is sort of more bare, right? There is stuff in there. It's just not a bunch of chloroplast. So that's one of the things that we're going to say in page 12. We're going to say both of those cells have a cell wall, a cell membrane, a nucleus, and cytoplasm. They both have roughly a rectangular shape, but Elodea had chloroplasts and onion did not. All right, and then we stopped there during class because we'll do more of those questions after we take care of the looking at our animal cells. All right, so there's an exit ticket for today, and you have 2 attempts. And then if you want, maybe I'll put this link for Magic School Bus Inside Ralphie. We watched just a tiny bit of it, and it wasn't really a main focus. It was just something that we had on as we were working. So thanks for checking in, and stop by for Falcon Focus if you have more questions or need to work on the microscope.